Hi friends, today we are going to study a new topic that is most important and interesting as well. Topic that is of biology. Okay, that is what? That is cell cycle and cell division. Okay, so like cycle. So what is a cycle? First of all, what is a cycle? We'll draw like a circle. First we are drawing. Let me give you a means basic. What is a cycle? First of all, this is a circle and this is a object. Okay, this is a object which is a living organism, which is the living organism. So this living organism will perform some of the biological cycle. Like there are many type of biological cycle. Okay, biological cycle. So like we are today discussing about the cell cycle or the cell division, you can say. Okay, and the next video will come on the curve cycle. Okay, okay, then there are many biological cycle are there. I will tell about it later on, but the next video will be on curve cycle. But today we are going to focus on the cell division. Okay, so let's move further. Okay, let me erase it first. Okay, so this living organism will perform a cycle. This is the cycle. It will perform many processes. It will go under many enzyme and hormonal change and many fundamental and physical change, and then it will come to the last stage of the cycle, that is coming like this. The cell will means the organism will go through a cycle. So this is the cycle. In this biological cycle there are many type of changes that occur. Okay, there is a change in fundamental properties, there is hormonal change and synthesis. Many biosynthesis takes place. Biosynthesis, let me write the name. Biosynthesis. Okay, so fundamental property, there is change in fundamental property, hormonal change, biosynthesis, and many secretion of what? Of enzymes. Okay, enzymes. Okay, and many other things in different types of cycle. So today's the biological cycle which we are describing or discussing today is the cell cycle. So let's begin the video. Okay, let me clear it out. Okay, now listen me carefully. Okay, so let me draw another circle. Okay, let me draw it more big and make it here. Okay, okay, see this cycle properly. We are drawing a line from here to here. Let me okay, let me make it as a red color and let me increase the thickness. Okay, so this is a line and let me draw another line here. Okay, okay, so first thing that takes place in the cell cycle. I am now starting the chapter. Okay, now the cell cycle will start. How the cell cycle will start? There will be a fundamental change before the real cycle happens. Means if I will tell about mitosis, how the division occur. A diploid cell is there. The diploid cell is dividing into two diploid cell. So we conclude that it is a equational division. Obviously it is a equational division. Okay, we are concluding as it is a equational division. Okay, like diploid cell. Okay, the diploid cell means the diploidity is of the mitosis is diploid. Okay, means the 2n is there. 2 means n the number of chromosome. Okay, 2 number of chromosome is there. Okay, Two numbers means if I am drawing the diploid cell photo like this, another copy means one copy is there, and here a different kind of chromosome. Okay, so this is basically the diploid cell. So this is basically the diploid cell. How it is diploid cell? That you can find one copy of chromosome of chromosome A, let's say. Okay, this is chromosome A, and if this is chromosome A, the next chromosome will also be A, because what I said, a one copy of chromosome A means this is A, and another copy of it again A. Okay, so if I am stating this chromosome as B, like the chromosome B, and the chromosome B have another pair. Okay, so there are one copy of chromosome, what, one copy of chromosome B, one copy of chromosome B. Wait a second. So here we can see that chromosome A is having one copy and chromosome B is having one copy. So total how much cell, how much chromosome? Total 2n. Okay, total 2n means it is diploid. N means the copies of chromosome, means here 2 into 1 means this completed. Okay, sorry, so there are two copies, means here is one copy plus here is one copy, so one copy plus one copy is equal to two copy, so 2 into 2 is 4, so here we are stating 4 chromosomes. Okay, so this is the mitosis. Okay, I have also told about it, the mitosis division. Okay, you can see my lecture videos, previous lecture videos. Okay, so let's move forward. Wait a second. So in the mitosis, first of all, means let's do not go to mitosis, first let's see the cell cycle. First thing is G1 phase, or we can also state that it is. So now I will focus on the G1 phrase, that is gap phrase. Okay, this is gap phrase. Okay, so in the gap phrase you will see that the cell is actually preparing itself fundamentally. Okay, so basically in the G1 phase the cell is actually preparing itself fundamentally. Okay, fundamentally it is preparing. What occurs in this G1 phase? We will see the DNA, the signals for the DNA replication is coming. Signals for DNA replication is coming. Okay, although DNA is not replicated in this phrase, DNA is not replicated, but the signals are coming for the DNA replication. Okay, then the next thing is that sorry, signals for the DNA replication is coming, but the DNA is not actually replicated. Okay, and the next thing is proteins are synthesized. Okay, RNA also synthesized, and what else occur? And the thing that the cell size grows, means here the cell size actually grows. Okay, means the cell is like this, and the G1 phase the cell will become like this. Okay, and the DNA will means the signal for the DNA replication will be also coming, and the protein and RNA also be forming, and the cell size is growing as I told. Okay, so next, and the next phrase is, okay, and the next phrase is, that is what, that is G2 G2 phrase I'm telling. S phrase means synthesis phrase. synthesis phrase, S phrase, or you can say synthesis phrase. It is basically, as the name states, that synthesize means it is synthesizing, means it helps in the duplication. Here the replication of DNA occurs. Chromosome, one chromosome is converted into two chromatid, sorry sister, sorry, I have here one sister chromatid is converted into two sister chromatids. Okay, two sister chromatid. Okay, so this is basically the S phrase. Okay, and here somehow protein are also formed. Okay, somehow the proteins are formed, synthesized. Okay, and RNA as well. Okay, and the next phrase is the what? The G2 phrase. G2 gap. Here the cell basically rests. Okay, actually telling rest is not good, because actually G2, in the G2 it is resting, although it is resting, but it have done many things, so it have to be rest. But here also what the wait a second, and in the G2 phrase it actually checks that DNA synthesized is sufficient or not. Okay, means more DNA or needed or not, like that. Okay, and here somehow some proteins are synthesized, some protein, not most, some proteins are synthesized here. Okay, synthesized here. And one thing I have left out in the S phrase, that is the replication of DNA. There is also the replication of what? Of what? Tell, you tell. Of what? Of centrosome. Okay, thing is that centrosome. Okay, if you will see the cell, means like this. Okay, this is the cell, and if the cell is not dividing, means here the cell is not dividing, here I am telling that not dividing. So if it is not dividing, then what is it containing? It is containing the nucleus. Okay, and in the nucleus it is in the form of chromatin, and the nucleolus also be there. Nucleolus, and you will see only one centrosome. How many centrosomes? Only one centrosome. Okay, only one centrosome. But when the cell is dividing, let me keep this here. When the cell is not dividing and comparing it to the cell is dividing, okay, and here the cell is actually dividing. Wait, here, and here the cell is actually dividing. Okay, cell is dividing means it is having chromosome. Okay, means here the chromosomes are been formed. Okay. Okay, here the chromosomes are formed. Okay, here the chromosomes are formed. Here you will see chromatin, and here when the cell is dividing, when the cell is actually dividing, you will see the chromosome, and you will see two centrosome. It's here one and here moving one. And in the meta phrase, which I will tell, it is a kind of phrase which occurs in the what, in the cell division, in the M phrase. I will tell like it will come to here, okay, this pole, and this will come to here. It will come in a central axis, it will come in a central axis, and here the division will takes place. Okay, means here the means this nuclear membrane will disintegrates, and this will form the spindle fiber. I will tell about it. Okay, and here the central axis means the central axis. The two centrosome will come to the central axis in which phrase? In M phrase. But in M phrase, which is the mitosis phrase, if I will go more deep in the mitosis phrase, in the chirokinesis and cytokinesis, in the chirokinesis there are many phrases. In that phrase, basically I am talking about the meta phrase. In the meta phrase, this will form the central axis, means the two centriole will come, centrosome will come to the central axis, and the cell division will further proceed. Okay, so this was the fundamental thing. Let's drive into our topic. Let's drive. Wait a second. Let's go. Now the cell have gone from the G1 phrase to S phrase. In the G1 phrase, what happens? The cell is preparing itself fundamentally, and the signals for the DNA replication is going, but the replication is actually not occurring. Actually the replication is not occurring, but the signal is going, and the protein and RNA formation also occur. Okay, protein and RNA formation also occur. The cell size also grow. Okay, so there is a growth seen in cell size in the G1 phrase. Then in the S2 phrase, it is synthesis phrase. As the name suggests, it is synthesis. So it will synthesize and means replicating the DNA and centrosome, which I have described now, and synthesis of RNA. Okay, protein and RNA, synthesis of protein and RNA. Then we proceed to the G2 phrase. What we have seen in G2 phrase, that it is actually resting, but it is not actually resting. Means it is resting, means we are telling it as resting phrase, but it is not actually resting, because here the DNA, means the DNA is checked that it is sufficient for the cell division or not, and some type of proteins are been synthesized here. Okay, some types, not all. Main protein synthesis occur in G1 and S phrase. Okay, then we proceed to the what? Then we will proceed to the M phrase. We can say it as meta phrase. We can say it as M phrase or meta phrase. Okay, we can say M phrase or meta phrase. What occurs here? Actual division occur, which I will tell about it. Okay, actual division takes place here. Okay, sorry. So today we will discuss about this M phrase. Okay, and this we have discovered all, means this G2, G1, S. Okay, so now just take a screenshot, or if you want to make a note, then make. Okay, my handwriting is bad, although, because I am now teaching, not making notes. Okay, so see this. Okay, so now I am rubbing it out. Wait a second. Okay, wait a second. Okay, you have done. Correct. See what occurs in actually here. Let me explain you. Okay, Okay, so if you will see this phrase, means particularly this part, let me draw it as green. This particular phrase means G1 phrase, S phrase, where the synthesis occurs of what? Of DNA replication and what? Centrosome, to this G2 phrase, that is resting phrase. So from this phrase actually means to G1 to G2 phrase, this all phrase means this G1 phrase, S phrase including the S phrase, and also ending at G2 phrase is actually what is actually interface, is actually interphrase, is actually interphrase, it is actually interface. Let me write it here. It is interphrase, and interphrase is the biggest phrase in cell cycle. In cell cycle, it is the biggest, means here the more time period is required in the interface. It is the biggest in cell cycle. Okay, then after that what occur? After that what happens? After that actual division occurs in this M phrase. Actual division. Actual, the spelling is wrong. Let me write it again. Actual division occur. Okay, so what we started? We have studied about cell cycle. In the cell cycle, we have seen what we have seen. We have seen the G1 phrase, S phrase. You can also state it as gap 1 phrase, synthesize phrase, G2 phrase. You can mark it as gap 2 phrase, and then M phrase. Here the actual division occurs. M phrase, that is mitosis phrase or meiosis phrase. If means it can mitosis, it can be meiosis. If it is meiosis, then here the two types of thing will occur. Means I am making it more easier for you. Like in the M phrase, it can be mitosis phrase. In the mitosis phrase, what we will see? We will see wait a second. We will see what? We will see two deployments. The diploid cell is actually converting into two diploid cell. Okay, two diploid cell. And in the meiosis phrase, what occurs? Meiosis phrase, what occurs? It is 2n, and the 2n is dividing into what? It is dividing into n and n. This is basically meiosis 1, and this particular divides into n, 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 and this part is basically meiosis 2. Okay, so here the meiosis occur in two phases, that is the meiosis phrase occurs in two phases, means 2 times the cell division will occur. Okay, meiosis 1, meiosis 2. Okay, so in the mitosis, in the mitosis, you will see what occurs. Basically, today's topic is on mitosis. Today I will teach only mitosis. Then the next class we will go for the meiosis. Okay, today I am teaching about mitosis. So in the mitosis, what occurs? Mitosis is further distributed into two types, means in the two phases. That is what? That is what? Chirokinesis, chirokinesis, cytokinesis. Chirokinesis, cytokinesis, and chirokinesis have five stages. Five stages. Sorry, four stages, I guess. Prophase, metaphase, anaphase, telophase. Prophase, metaphase, anaphase, telophase. Okay, and then at the last the cytokinesis occur, where means the chirokinesis means nuclear division. Nuclear division. Here the nuclear division occur, and in the cytokinesis, here the cytoplasm division, cytoplasmic division occurs. Okay, so this was the two things. Okay, so today I will give, means tell about it more detailed by drawing diagrams. Okay, so let's move further. In the mitosis, you will see, like here we are starting the mitosis part. Mitosis, what occurs in the mitosis? Here we are not talking about only mitosis phase. I am talking about the whole mitosis process. What occurs? First the G1 phase comes. G1 phase comes. Here what occurs? Here what occurs? Here signals for DNA replication. Actual DNA replication doesn't occur. Signal for DNA replication. Okay, signals for DNA replication. Okay, then synthesis of proteins and RNA. Protein and RNA occur, and then what occurs? Synthesis of protein, RNA, and signals for the DNA replication occur. Okay, then here is seen cell growth, means the cell is growing, means the cell is like this, and after the G1 phase, after the G1 phase, means in the G phase it is becoming like this. Okay, so there is a growth seen in the mass. Okay, then we are going, what occurs, what else occurs? I have forgot about it. Let's see here. Okay, I have deleted here. Okay, no, the signals for the DNA replication occur, synthesis occur. Okay, the growth is also seen. Okay, okay, and here basically it is preparing fundamentally, it is preparing, yes, it is preparing itself fundamentally for division. Okay, so this was the thing. Okay, next we will see what we will see G2 phrase. Okay, I am making it as green. Okay, then we will see what? Then we will see the G2 phrase. Sorry, the synthesis phrase, that is S phrase. Then we will see the S phrase. What occurs in S phrase? Here, synthesis of protein and RNA occur. Replication of DNA replication of centrosome, replication of a centrosome, synthesis of protein and RNA, then replication of DNA, then replication of centrosome. Okay, one centrosome was there, but after the S phrase, you will find two centrosome. Although the centrosome are not fixed in the central axis, in the metaphrase, that is in the chirokinesis phrase, it will come in the what? The central axis. Okay, the replication of centrosome. Okay, so this was the thing. Okay, these occurs. Okay, these are the means that occur. Then what comes? What then comes G2 phrase. Then comes what? Then comes G2 phrase. This is actually the resting phrase, but it is doesn't resting. Okay, I am writing resting phrase, but actually it is not resting, because it is investigating the DNA, whether it is sufficient for division or not. And some proteins are formed, some, not all, means not most. Some proteins are synthesized, some proteins are synthesized here, some protein are synthesized here. Then we have studied about the G1 phrase, S phrase, G2 phrase. Then we are going to metaphrase. Sorry, metaphrase I'm talking. What a fun fact. I'm talking about M phrase. And basically, if we are studying about mitosis, this M phrase is actually what? The mitosis phrase. And in this mitosis phrase, the actual cell division occur. Actual cell division means, here I am talking in the M phrase, what occur? Chirokinesis, then cytokinesis. And in the meiosis, actually in the meiosis, it is the mitosis. But if I am talking about meiosis, then there will be chirokinesis 1, cytokinesis 1, then again chirokinesis 2, then again cytokinesis 2. Okay, so this we will study on next class. Okay, and in the next class also coming the interesting topic, that is Krebs cycle. Okay, we will also cover it. Okay, so here see, chirokinesis, cytokinesis. In chirokinesis what occur? Prophase, metaphrase. Okay, I am writing like this. Prophase means P, metaphrase, anaphrase, telophrase. Okay, PMAT. You can also go for what? PMAT. This is the PMAT phrase. Okay, you can also go for like this, PMAT. And here the simple cytoplasmic division occur. And this is actually the nuclear division or nucleus division, you can go for it. Okay, so basically we are starting from the chirokinesis 1. So when the cell is not dividing, like here, if we are talking about this cell is actually not dividing, as we can see that it is having the what? Having the chromosome. Here we are not describing about other cell organelles. We are only focusing on the what? We are only focusing on the nucleus and centrosome. Okay, So these are the two things, and the most important thing, chromosome. Okay, so this we are stating the means endoplasmic reticulum, the Golgi apparatus. It is not needed for now. Just see what I am telling. Now you will see that it is chromatin. It is what? It is chromatin. And here you will see one centrosome is there. Means one centrosome is present. After completing of the interphase, this whole is actually the interphase. This whole phrase is actually stated as inter interphrase. Okay, this whole, the G1 to G2 phrase. Okay, okay, after this phrase you will see that the chromosome is converting like this. Means here the DNA is replicated, means here the DNA is, some amount of DNA is there, but here in this cell you will see the DNA is replicated, means more amount of DNA you will see here. Okay, you will see more amount of DNA here, and what, and two centrosome which are not in the central axis. Okay, here you will see more amount of what, DNA. You will see here more amount of DNA and nucleolus. This is nucleolus in red color. I am telling nucleolus. Okay, here it is about the interphrase. This is the interphrase, sorry, the, okay, it is about what, it is about interphrase. But when it is starting the real mitosis, okay, the first phrase we are talking, that is the chirokinesis, and in the chirokinesis first we are discussing about the prophrase. So what occurs here? Here the nuclear membrane disintegrates, nucleolus also disintegrates. Okay, and what else occur we are describing here. Like here you will see, and the most important thing is that the chromatin is converted into chromosome. Okay, these are the things that occur in the prophase. Okay, so if I will say this cell, so like this is a cell. Okay, you will see that it is having nucleus. Okay, so the nucleus will disintegrate. Okay, nucleus will slowly pop out. It is disintegrated, and then the chromatin which you are seeing is actually is not now more chromatin. It is now what? It is now chromosome. It is now what? It is now chromosome. It is now what? It is now chromosome. It is what? It is now chromosome. Okay, so now you will see this, and the diploidity which you are seeing here, the diploidity you will see here as 2n. Okay, this is the 2n cell. Means if I am wait a second, I am writing it as 2, means I am writing it as 2n. Okay, this 2n. Okay, and here the centrioles are present. But it is not in the central axis. Okay, Okay, so these are the things that occur. Okay, then we are moving to what? Then we are moving to prophase, metaphase, anaphase, pmat. I have told what? pmat. Okay, so this was the what? This was the prophase. Okay, we are moving to metaphrase. We are moving to metaphrase. What occurs in the metaphrase? Let's see. In the metaphrase, the spindle fiber formation starts. Spindle fiber formation from centrioles. Now we are not writing centriole, centrosome. Let me write centrosome. The spindle formation from centrosome occurs. Okay, and the two centrosome are coming and aligning at central axis. So here you will see, if like it is not in the central axis, means this is not in like this position. Okay, so it is not in like this position, but in the metaphrase, which I was telling the beginning of the session, in the metaphrase the central axis will come. Okay, the two chromosome will be at central axis. So let me draw like this, and oh, sorry, I am not drawing a proper okay, let me do okay, we can duplicate also here. Duplicate. Okay, this is very simple. Okay, so we can duplicate. So now we see that here the spindle fiber formation started, means the spindle fibers are actually forming. Okay, it is actually forming. Okay, the spindle fiber is now what? Forming. Okay, and you will see here the what? Chromosomes. Okay, you will see the chromosomes at here. Okay, then what occurs in the chromosome? If you will understand the structure, let's move to the anaphase. I will tell in the anaphase. Okay, you have seen the spindle fibers are forming. Spindle fiber are forming. Okay, so you might be wondering what is actually in the centrosome. Let me describe about it. The centrosome is actually looks like this, means this part I am enhancing here. Okay, I am enhancing here. This part I am making it, this part, once again, this part I am making it here, and if I will zoom it, means this part, now the central fiber has formed. See this. These are what? These are the two centrioles, which is at perpendicular axis. It is obviously, you are seeing that it is perpendicular, that is 90 degree. You are seeing that it is in the 90 degree. So in one centrosome, the spelling is wrong. In one centrosome, you are seeing two centrioles. What you are seeing? Two centrioles. So this is the thing. Okay, and how the spindle fiber is actually formed, that I have not discovered that. How it occurs? Centrosome In the centrosome, this part is called what? Centrioles. Centrioles are formed by using what? By using microtubules, and the microtubule is formed by using tubulin protein. It is a kind of protein. So this tubulin, the presence of this tubulin protein in the centriole helps in the formation of what? Of spindle fibers. Like this, the spindle fibers are formed. Here I have explained only briefly. Okay, let's move to the anaphase. Okay, let's move to the anaphase. Okay, in the anaphase, what you will see, the big split of chromosomes. Two nucleus are not formed. Means, if we are thinking like this, the centrosome will come towards it and the centrosome, no, two nucleus are not formed, and what else? And the diploid chromosome comes towards centrosome. Means what occurs actually, let me duplicate it. I am explaining. Okay, so let me explain you here what we are seeing here. The spindle fiber formation was going on. Okay, now the big split will occur. Means the centroles are here, the centrosomes are actually here. Okay, and the spindle fiber is actually, means the chromosome was like this, no, means the chromosome was like in this shape. Okay, here the sister chromatid segregation will occur. What will occur? The sister chromatid segregation means this, like this. Means if think like it is what I have told you earlier, that it is a diploid cell, because two types of copies you are seeing. One is this copy and one is this copy and one is this copy. Okay, so two type of copies you are seeing. Wait a second. So it is what? 2n diploid cell. So it will lead to formation of two diploid cell. I have told it is called mitosis. 2n and 2n. So here these are known as sister chromatid. Means if I will tell the structure of chromosome like this. Okay, let me draw it more better. Okay, if I will talk about the chromosome. Okay, so these are the chromatids. So in one chromosome you will see how many chromatids? Two chromatids. How many chromatids? Two chromatids. So in one centrosome you will see one chromosome you will see two chromatids, and in one centrosome you will see two centrioles. So this was all the concept. Okay, let's move further. Okay, so now you have seen. So what actually occurs here? The centromere is there. It is what? It is called centromere. Let me erase it. You have studied that it is chromatid. It is one chromatid and this is two chromatid. So here it is what? Two chromatids. Now it is centromere. Wait a second. Now it is a centromere. In the centromere you will see kinetochore. You will see what? here you will see kinetochore. Kinetochore. What is this kinetochore? Here actually what occurs? Here actually the spindle fiber is attached, means like this. In this part is actually the centrosome, means in this kinetochore the spindle fiber will attach to it. So basically here the spindle fiber will attach to what? To attach to the kinetochore. These are the kinetochore and this is the centromere. Okay, so here the pull will occur not from the DNA. If you are thinking like this, from here the spindle fiber is pulling out, no, you are totally wrong. From here it is occurring. Okay, from the what? From the center, from the where? From the centromere, and in the centromere specifically in the kinetochore. Okay, correct. Okay, let's move further. So I am erasing out because I was describing about it. You have learned. Okay, let's move further. Now the pull will occur. Like the pull will occur means what? Here you will see that 1 2 1 2 1 2 means here it is 1, means 2 copies were there, no, like this. So what will actually occur? This part will go to here, okay, and this part will go to here. Okay, and this part will go to here again, and this part will go to here again. Okay, and this part will go to here again, this part, and here also 2 chromatids there now. So 2 chromatid, here is 1 chromatid, okay, and here is 1 chromatid, and here also 2 chromatid. So 1 is here, and 1 is here, and 1 is here. So it is what? So it is what you are making? You are making 2n converting into what? 2n means afterward it will replicate. Okay, afterward it will replicate and become fully 2n. Okay, let's see what occurs here. You have pulled out the what? You have pulled out the chromosomes. Okay, so here a non-kinetochore spindle fiber will be there. Non-kinetochore spindle fiber, like this, means it is attaching to the kinetochore. This spindle fiber means the red color. Okay, I am marking it as red color. The red color ones is attaching to the kinetochore part. So this spindle fiber, this type of spindle fiber will be termed as a kinetochore spindle fiber. What? Kinetochore spindle fiber. But this green color part I am labeling at. This green color part is actually non-kinetochore spindle fiber. Non-kinetochore spindle fiber. So what actually occurs here is that it actually lengthens, means this fiber, this non-kinetochore spindle fiber is actually lengthening up means it is stretching actually, like this. It is stretching means it is like this, no, so it is stretching itself and making large. Okay, so it will large, then it is sorting, means it is lengthening, but it is sorting. Obviously it is sorting, means this part is coming towards it, means reducing. And this spindle fiber, this red color part, it is reducing, no, it is coming back to its position, so it is reducing. So it is reducing the means the kinetochore spindle fiber, but the non-kinetochore spindle fiber is actually lengthening up. Okay, so after this the chromosome will come to like this. Okay, like this means this will come to, okay, I am making another duplicate it just like this. Okay, this will become like something like this, and it is pulled out. After pulling it, it will become in the chromatin form. Okay, it will become the chromatin form, and the non-kinetochore spindle fiber will disintegrate slowly. It will disintegrate like this. It will slowly disintegrates. It will slowly disintegrate. Okay, then I am talking about the final step, that is the fourth step. That is what? That is telophase. In this telophase, what actually occurs? Actually here no, here the chromosomes are in the chromosome form only. Means here the chromosomes are like this, means the chromatin is not formed. But in the telophase, the two chromatins which are pulled, I am writing like this, the chromosome will convert again into chromatin. Okay, will convert again into the chromatin, and two nucleuses will form. Two nucleus will be formed. So here in the telophase, what occur? I am making another duplication. Okay, duplication. Wait. Nucleus, what is formed? Like the centriole are there here, and here the chromatin, means this chromosome is converted into what? Chromatin. Here the chromatin is formed, and what else occur? It occurs nucleus, means here the nucleus membrane occurs. Okay, here the nucleus is formed, and then the inside part, that is the nucleolus will also forming, and after this phrase the total what? The total chirokinesis is over. Now we are moving to what? We are moving to cytokinesis. What else occur then? What else occur? This will converted like this, means here will the pressure will generate, means here the dips will generate the depression. Here the depression will generate like this. Okay, here the centrosome, and here the depression is actually formed. Okay, the depression is actually formed. Here the pressure will be there, and finally two cells will be generated successfully. Two cell will be generated what? Successfully. Okay, like this things occur. Remember one thing, mitosis is actually growth. Mitosis is for development purpose, it's for growth purpose. Okay, growth purpose. It actually occurs in the germinal cells. It occurs in the germinal cell. If I am talking about the what, in the male, it is basically the primary spermatogonia. But in the female, you will see it as primary oocyte. Okay, so these were the things, but it doesn't occur in the germ cell. There is a difference between the germinal cell and germ cell. Germinal cell means the primary spermatogony and primary oocyte, but the germ cell are the sperm and ova. Here the mitosis doesn't takes place. Okay, here only the what the meiosis takes place. Okay, the meiosis, and it also takes place in the gonads. Gonads means the reproductive part. Okay, so and occurs in the somatic cell also. Somatic cell, here actually the sister chromatid segregation occurs. Okay, in the mitosis basically sister chromatid segregation occur. Segregation occurs. Okay, so this were the fact. Okay, revise it all which we have studied. Then at the next class we will go for the meiosis phrase. This is the most interesting and the long phrase, and then we will go for the most interesting, that is Krebs cycle. Krebs cycle is difficult, but we will complete. Then we will go to C2 cycle. C2 cycle, which is the photorespiration. And before discussing the C2 cycle, we will also discuss about the what the microbodies. There are two types of microbodies, that is peroxism, that is what peroxism, and the second is glycosome is basically the specialized peroxism, but it works different. In the C2 cycle, actually the loss of CO2 occurs. We will study about it. It is actually the 10th class topic, the C2 cycle. But the Krebs cycle and this cell division is actually 11th 12th level, but in the 9th class also it is there. But in the 9th class the proper definition and the description is not described like this, which we have studied. Okay, so bye guys, bye. We have taken the lecture for how many minutes? Let me see. Okay, so 1 hour 2 minutes. Okay, so today's lecture is, and we have done a record of our YouTube channel, that is 1 hour 2 minutes video. Okay, we have made. Okay, bye bye guys, revise it all. We will meet again. Okay, so bye guys, bye.